Thanks very much for joining today. I'm sorry I can't be in Chicago for those who are there, but the Zoom sessions seem to work reasonably well. And what I wanted to do to start off this session was talk about two things. One is this question about how is it that GFAP variants cause a leukodystrophy? This is a gene that's not expressed in oligodendrocytes, the myelin-forming cells. So how is it that these GFAP variants lead to leukodystrophy? And the second part of my presentation is going to be online sources of reliable information, for the most part free as well. So hopefully those will be useful to people. So let me just start out with this first question. How do GFAP variants lead to leukodystrophy? So, we've known for 20 years that the genetic cause of Alexander disease are variants in this astrocyte intermediate filament protein called GFAP. So in some way, these GFAP variants lead to astrocyte dysfunction. That part of the story is pretty clear. The end result is pretty clear as well, that there are myelin deficits as well as other aspects of the Alexander quote unquote phenotype. But what the major unknowns are how you get from astrocyte dysfunction to the myelin deficits. What exactly goes wrong in the astrocyte that leads to problems in myelination? And, I was struck by the presentation on Thursday morning by Roger Min from the Netherlands group, talking mostly about another leukodystrophy, MLC, but in addition, he put it into the context of some other disorders, all involving some abnormality of ion or water regulation in the central nervous system. And that's really interesting because I think there are emerging hints that that may be part of the story for Alexander disease as well. And in particular, I'll highlight these three genes which encode proteins, KIR 4.1, which is a potassium channel, connexin 43, which is a gap junction protein, and aquaporin 4, which is a water channel. So each of these are proteins that form channels in the membrane and allow either ions or water to go through. And clearly there are genetic disorders where these proteins are the primary target, and you can have, at least for KIR 4.1 and connexin 43, and you get myelin deficits. Aquaporin 4, there's an autoimmune disorder that involves inflammation in the brain. And something similar to that may be happening in Alexander disease. The evidence for that is coming from our new rat model that was just published last year. And this is on the right-hand side. By the way, can people see the cursor as I move it around, or is that just me? Oh, you can, okay. So this is a Western blot for those of you who aren't familiar with these. These are proteins that are run along a lane longitudinally. So we're looking at 4 animals here, wild type animals in these 2 columns, 2 Alexander disease rats in these 2 columns, brainstem samples here, spinal cord samples here. And in GAPDH is the bottom is a control protein that shows that equal amounts of protein were loaded in each lane. But you can see for each of these 3 proteins, connexin 43, KIR 4.1, and aquaporin 4, there's a substantial reduction in the amount of the protein that's present in brainstem and spinal cord compared to the wild type animals. And so here, right here, it's suggesting that some abnormality of ions or water regulation in the central nervous system might be a common theme among several of the leukodystrophies, including Alexander disease. And exactly how you go from the variants in GFAP to these changes in ion channel proteins, we don't know yet, but it's something that we're going to be following up on quite a bit. Of additional interest is, of course, the fact that what we spent a lot of time working on that was published in this paper as well is whether you can correct any of these deficits in the animal models of Alexander disease through treatments such as antisense therapy. If I just go back to this slide for a minute, one of our problems is that we are identifying so many different things wrong in these animals that it's a little hard to figure out where to start in terms of a rescue. Do you replace connexin 43, KRF 4.1, aquaporin 4? How many of these things do you have to fix in order to really provide a meaningful treatment? And the strategy we've been pursuing for several years is that all of this would be taken care of if we just get rid of the GFAP. And that's what led to the experiments with Antisense. And this is a figure also from that rat paper published in 2021 for KR 4.1. Here you can see the Alexander rats here, much lower KR 4.1 compared to the wild type animals. Aquaporin 4, same thing. These are animals treated with phosphate buckwheat saline as a control, but those animals treated with antisense show a substantial rescue, essentially back to control levels. So it's these kinds of findings from the animal models that has really motivated moving from the lab bench into a clinical trial, and has us really excited to see, first of all, that the clinical trial is underway, and eventually we'll find out the results. I think we're all very convinced that this works very well in an animal. We just don't know what's going to happen once we move into human. Okay, so let me move on to the next part of the talk, online resources. And the web is a dangerous place to be because you can find lots of things out there, and some of them are old and they're inaccurate, and they really can get you very confused if you don't have some sort of a guide. So that's what I tried to put together here, online resources that are for the most part free. The first one I'll mention is this book that I put together. It was first published in 2018, an updated version with a new appendix in 2021. And this is a guide for patients and families. Other people may find it as of interest as well. And if you just go to this website at the bottom, you should be able to download that for free. I'll give you all of these links again at the end, and I believe the ULF is recording this anyway, so you'll be able to come back to it. The second thing for people who are interested in wading through the original literature is that for several years, I've been putting together this research bibliography. And you go to our website down here, and one of the tabs is labeled Comprehensive Research Bibliography. And if you click on that link, you get to a Word document, which again, you can download. And I update this continuously whenever something new comes out, or in this particular case, I realized there was a paper from 1966 that I really should have had there, but didn't. And so that's now on there. This was the first time anybody had done electron microscopy of Rosenthal fibers in Alexander disease. But I put virtually everything that's published on Alexander disease into this bibliography, even things that really don't provide any meaningful contribution. And when I feel pressed, I add editorial comments like the one up here. This is a very misleading paper. There's a lot of mistakes in it. And to me, Alexander disease shouldn't even be in the title. So it's there. And for that reason, people may think that there's some value to it and want to go look at it, but I wouldn't bother. There's another part that I also include, which starts off with just some general overviews. I already mentioned the book. It provides the link to getting a full text. This provides an overview on essentially every aspect of Alexander disease, except symptom management. And I left that in the hands of the experts. So unfortunately, Amy Waldman and two others published an article in 2020. This is an update of an earlier one on gene reviews. And this review does focus much more so on the clinical aspects of the disease. And again, you can get to that through a full link. In fact, if you click on this link here, it takes you to the National Library of Medicine bookshelf page, and somewhere on this page you click and you can download that article. Once you get, I should say that in addition, I've identified, following this box with the reviews, I have a section on what I think are the key publications going back to the beginning. The first paper by Stuart Alexander in 1949. Not calling it Alexander disease. Of course, that didn't come until later when somebody else decided to name it after him. And I had complete control over this. So I decided to identify 19 papers that were the most important ones going back to the beginning. And then following that, there's the reverse chronological listing of everything. It's about 50 pages long now. There are almost 400 articles. Pretty much everything is there. If you go to one of these, let's say this one, and click on that link there, the title is linked again to PubMed. And for those of you who aren't familiar with PubMed, this provides the journal, the year, volume, pages, title, authors, all that. And in addition, it'll show you a summary of what's in that paper. If you're at a place that has access, through a library, for instance, to a journal subscription, then you could download the whole journal. If in this particular case, you don't get that for a couple of years, but there are increasing numbers of journals that are open access, and I'll be providing the links to that full journal article in this bibliography. The next thing is an email list. There are maybe 120 people on this email list right now. Once a month, I send out listings of new publications and whatever other news there might be. If you want to be added to that email list, just send your email or send me a note to my email address, which is right here. It's very simple to unsubscribe yourself from it if you don't find it useful. The next things are podcasts. We started this, I guess, maybe almost a year ago. And once every month or so, I and a few guests get together to talk about some of these recent publications and sometimes some other topics as well. So this just shows the last two. Episode 10 just got uploaded earlier this month. We talked about Natasha Snyder from the University of North Carolina, Chapel Hill. And I talked about a paper that she was a co-author on that looked at 14 different variants in GFAP and tried to understand how those variants affected GFAP assembly and aggregation. And then also Natasha presented some new ideas about functions of intermediate filaments. So these aren't on any regular schedule. It's whenever something comes up that's worth talking about. And I have a guest lined up. And then last, we started having Instagram, Facebook postings of what I call a data slide of the week series. So on each Saturday, I put up a figure from a recent paper. And right now we're just working our way through the rat model from my lab. And so it'll show that figure as well as a brief description of it. And so this is a hopefully easy way to go through these kinds of publications in bite-sized chunks. And of course, anytime people have questions about it, they need to write to me and we can discuss them on a podcast. So with that, I've put all of those different websites together on this one slide. Again, I hope that the ULF is going to be posting, well, certainly they're doing the recording. I don't know how they distribute that later on. Perhaps I could ask them to post some of these things directly on their website going forward, and people can use that as a guide. So thank you for listening. I don't know if you want to do any questions now, or we just save them for the end.
All right, thank you. So both online and in the room, are there any questions? I'm just looking around. I don't know if you can see me on camera, but we've got folks just kind of taking it all in. And I'm going to turn around and see if we've got any coming in through the chat. If you guys want to send any messages, questions, pardon me.
Dr. Messing, I've been monitoring the chat. I didn't see any questions particularly for you.
Okay.
Thank you.
All right. So I can stop the share.
You can, and thank you very much. All right, I'm going to pass the baton.
Thank you. I'm gonna. I put that there. Can you hear me online? Yeah. Okay. Perfect. Okay, so hello everyone. My name's Katrina Moore, and I work in clinical development at Ionis Pharmaceuticals. And I'm delighted to be able to talk to you today, just to give you a refresh about the Alexander disease clinical trial. So I'm going to start today just by telling you a little bit about Ionis. I know a lot of you are probably familiar with us, but just in case some of you aren't, just to give you a little bit of background. Then I'll talk a little bit about antisense oligonucleotides, and then I'll remind you of the design of our clinical trial. So Ionis was founded in 1989. It was originally called Isis Pharmaceuticals, and as you can imagine, we changed our name. And we're located in Carlsbad in sunny California. And so we're the leading innovator of RNA-targeted therapeutics, which I'll talk to you about a little bit more in a moment. Ionis is made up of world-class scientists and leaders, and we have a passion to deliver transformational medicines to patients who need them. Ionis is largely focused on the research and the development of antisense medicines, which are designed to disrupt the disease progress. We have a number of different drug programs in development, and an example of 3 approved medicines includes Spinraza or nusinersen for SMA, and also Tegsedi and Waylivra for 2 other rare indications. So we were formed as a company to do this one thing, this one type of drug, and we have multiple drug programs in development with a strong emphasis on the treatment of neurological disorders. And as you can see from this list here, we have a lot of drugs already in the clinic and many more that are in preclinical testing or in research. So we're really excited to test gfap targeting antisense oligonucleotides as a possible treatment to Alexander disease. And I just wanted to talk to you briefly about how ASOs work. And so to do this, the first place that we need to start is to think about how proteins are made. And this starts with DNA, which you can see pictured on the left. The DNA is the genetic material in your cells. And so you can think of DNA really as like the master cookbook to help make all the proteins that your body needs. And so each protein has its own unique recipe, and this is called a gene. And DNA is the cookbook for all of those genes. Now your cells are able to make millions of different proteins, and proteins are tiny molecules that are required for nearly everything related to how cells live and work. For example, they serve as structural building blocks. They're responsible for waste cleanup and routine maintenance. They receive signals, they send signals, and they also mobilize response to viruses. Every cell in your body contains tens of millions of proteins at any given time, and they're all working in their own way to help keep you healthy. So we're really heavily dependent on our ability to make the proteins we need in the quantity we need when we need them. So DNA naturally exists in a tight double helix structure in a special compartment of cells called the nucleus. and being double-stranded and protected in the nucleus helps to keep it safe. After all, it's this master cookbook, and so it's really important that DNA is well protected. Now, when your body needs a protein, the DNA unwinds a little bit to expose the recipe for that particular protein, also called the gene. Now, this is where the really interesting thing happens. A temporary copy of the recipe is made, and this copy is called messenger RNA or mRNA. Now, the mRNA is much more mobile than DNA. And so this mRNA copy of the recipe is able to leave the nucleus and interact with the cellular machines that read the mRNA and use that information to help produce a protein. So this is how the gfap protein is made. There is a tiny area in the DNA that holds the recipe for making gfap. When your body wants to make gfap, it first makes a copy of the recipe. That copy, as we know, is called mRNA. And then the mRNA recipe for gfap goes off into the cellular machines which produce the gfap protein. So Ionis developed a technology called Antisense Oligonucleotide, or ASOs, and this type of drug acts on RNA to affect the amounts of proteins that are made. So let me show you how this works. So here is the same process that I just showed you. So we have DNA at the top, and then the mRNA copy is made. It's the mRNA that is destined to leave the nucleus and be used to produce a protein. Now, ASOs can be designed to work in dozens of different ways. And on this slide, I'll show you the most well-studied ASO mechanism. This is the way to use an ASO to lower the amount of a particular protein that's made. It also happens to be the ASO mechanism that we're using in the Alexander disease program. So what we do with antisense oligonucleotides is we design this tiny little molecule shown here by the purple strand, which you can see, maybe not. Anyway, you can see it in the top corner. And so the purple molecule is made of chemically modified DNA, and we design an ASO so that it will recognize the mRNA of interest. When the ASO binds to the mRNA, it creates this duplex, which is shown by the orange and purple strands interacting with each other. Our bodies are designed to recognize errors in genetic material. Now, double-stranded DNA doesn't raise any alarms. After all, that's exactly how you would expect it to be. It turns out that double-stranded RNA also doesn't raise any alarms. RNA is usually a single strand that floats around, but it's also okay if it's a double strand. But when RNA binds to something that looks like DNA, which is what we have here with this orange and purple mRNA ASO duplex, that's when the alarms sound. Your body recognizes this as an error, and it turns out that your body has a really clever mechanism to deal with this error. Now, a naturally occurring enzyme called RNase H1 finds the RNA duplex and destroys it. Specifically, the RNase H1 cuts the mRNA, which you can see here represented by the scissors. And after the mRNA has been cut, it can't be used to make any more of a protein. So with ASOs, we are tricking the body into cutting up the mRNA message, which should result in less protein being made. Now, as I note here on the right of this slide, ASOs are highly specific and titratable. By highly specific, I mean the ASO is designed to interact with a particular mRNA. For example, in Alexander disease, we're interested in lowering the amounts of gfap protein that's made. So we've designed an ASO that will only interact with gfap mRNA. By titratable, I mean that if we administer a small amount of ASO, there'll be a small amount of lowering in the protein. And if we administer a large amount of ASO, it will cause a larger magnitude of the change of protein. So it's important to understand that ASOs can be treatments, but they're not going to be cures. Like any other drug, the ASO will eventually be cleared from the body, so the ASO's effect on the protein levels only lasts for as long as the ASO stays in the body. So now let's talk about the possibility for the ASO for the Alexander disease program. Now, in Alexander disease, cells make too much of the GFAP protein. Everybody has 2 copies of GFAP, just as you have 2 copies of all of the other genes in your body. In the case of Alexander disease, there's this tiny little change in the DNA for one of those copies. We call this change a mutation in the gene. When you have this mutation, the gene, the GFAP, it results in what's called an overexpression of the gene. And that means that too much GFAP protein is made. So as I mentioned, we've designed an ASO that will target the GFAP mRNA, and using the mechanism that I just showed you on that previous slide, the ASO binds with the mRNA, which causes it to be degraded. And if the GFAP mRNA is degraded, it can't be used to make the GFAP protein. So we'd like to use this approach to treat individuals with Alexander disease because the underlying of the cause of the disease is the mutation in the GFAP gene. Just to note, the ASOs don't change the DNA, so the mutation remains in the DNA, but we'd like to administer the ASO so that it intercepts with the GFAP mRNA and leads to its destruction. So let's talk about the program itself. So we started in around 2014, 2015 with the idea to design an ASO that targets GFAP mRNA. And so we went through this process shown here at the top by the long arrow. And I think what's important to highlight here, as you can see in this arrow, is that it really doesn't do justice to the magnitude of work that is involved in a research program like this. So first of all, we started out by looking at an ASO that targeted the mouse version of GFAP and also the rat version of GFAP. And we looked into the concept of lowering GFAP in animals to see if it's potentially beneficial. Then we moved on to designing an ASO that specifically targets the human GFAP mRNA. And we selected the one that we thought looked like it had good properties to be able to make it into the clinic. After we found the most promising ASO, we embarked on a lot of testing in animals, which you have to do as part of clinical development to be able to test the potency and the tolerability of the drug in animal species before moving it into humans in the clinic. So as I mentioned, the program itself started back in 2014 with a really wonderful collaboration with Dr. Albie Messing, who you just heard from. Now, he reached out to somebody at Ionis after he saw a presentation about ASOs and said he thought it could be helpful for Alexander disease. And so Dr. Messing had been working for a long time on the foundational research of Alexander disease to really understand the mechanism. And at this time, we collaborated, and Ionis brought its 30 years of experience developing ASOs to understand how to test them. And so to get to the point where we are today, we had to screen thousands of ASOs to identify the one that triggers the destruction of GFAP mRNA. And the drug that we identified and is currently in the clinic right now is called ION 373. And so we're collaborating with expert physicians around the world, including Dr. Amy Waldman, who's speaking after me in a moment, to help support a successful program. And at the start of this month, in June, we started enrolling patients into the second cohort of the study, which is called Cohort B. So here on this slide, I just wanted to summarize for you the Alexander disease clinical trial design and where we are in the study, as I just mentioned. So I think one important thing to remember when we talk about clinical trials is that they're research. They are experiments. And when we design a clinical trial, we have a number of questions that we're looking to answer, such as, is this ASO safe? Is it beneficial? Do people with Alexander disease benefit from taking our medicine? And so we've designed an experiment to help generate data to help answer these questions. Another important thing to think of is that really we're at the very beginning we're not at the end of our experiment. And so we're at this stage where we can learn about the possibility of an ASO as a treatment for Alexander disease. So in our trial, we're looking at enrolling patients with Alexander disease. And sometimes the first trial in a new drug is in healthy volunteers. But that's not the case here. We're going straight into individuals with Alexander disease. And so study participants receive the study drug every 12 weeks. And they will either receive 10 doses of Ion 373, or they receive 5 doses of a placebo, followed by 5 doses of Ion 373. So eventually everybody ends up on the treatment. Patients, parents, staff, and also us here at Ionis are mapped to the treatment assignment. So the study drug is administered intrathecally into the CSF via a lumbar puncture. So that is an injection into the base of your spine. We have a broad age range in the study from 2 to 65 years old, and we're asking that patients be ambulatory if they're 5 years of older. And importantly, we require patients to be able to safely participate. In the study, we have endpoints in the trial that include performance tests, questionnaires, physician reports, lab tests, and many other things. And so really it's a long set of assessments, but at the end of the study, we use all of the tests that are administered to measure changes in motor function, cognition, communication, and everyday functioning. And we also assess the safety and the tolerability of the drug. And so we have several clinical sites all around the world participating in this trial, and we're aiming to enroll approximately 42 participants. If you'd like to learn more about our Alexander disease program, you can visit our website at ionistrials.com. You can also look at clinicaltrials.gov. That's a great resource for information, and you just need to search the term that you can see here. And you can also always reach out to our patient advocacy group here at Ionis by emailing or calling that number that is shown on the slide. So thank you so much for listening. I'd like to give you the opportunity to ask any questions, if there are any. Okay, hang on. I'm gonna just connect to my microphone.
Passing the mic. We're not passing the buck by any means. So first, while we're pulling up some online questions, I'm going to turn around to the room and see if we do have a question, and I'm going to excuse myself to get closer. Why don't you go ahead and ask it and we can repeat it.
Right. And yeah, so why is that the frequency of administering the drug? And based on the results, do you anticipate that it could change?
And then we've got an answer.
Oh yeah, that makes sense.
Yeah.
Okay, so the question in the room was, why is the interval determined? So why are we giving the drug every 12 weeks? And then also, could that interval change at any point? Is that? Yeah, for 2 years. Yeah, so a lot of the work is done in the preclinical models. And so we do a lot of modeling to be able to determine, so we look at how much GFAP can be knocked down in the animal models, and we see how long we can maintain that knockdown for before being able to administer another dose. And so that's why every 12 weeks was determined. As we work through the study, we look to see, because this is all done in animal models, and sometimes that's a little bit tricky to figure out if that translates to humans. So we're just doing the best approach that we think. If the data looks good and it looks like we're maintaining the GFAP knockdown, and it looks like we might be able to increase the interval, then we will. Also, it might be that we're not achieving it every 12 weeks and we have to give it more frequently. So right now, 12 weeks is what made sense based on all of the information that we have, but it can always change in the future once the human data comes in from the trial participants. Yeah? Yeah. Yeah. It would likely be for the rest of somebody's life. It's just you set an initial interval to be able to evaluate the time course of the drug and get all of the information that you need. So that's why the length of the trial is set. Any other questions? Yeah. So the question was?
Question is, I understand that the trial was due to last about 3 years. Has this time started yet, or will it not start until the last participants are enrolled?
Yeah, so the length of the study starts when the first participant is enrolled. And then the duration of that study is predicted to be 3 years based on how quickly we think we can enroll participants. But it really depends on when that last participant is enrolled based on how long the study will go. But right now, we think approximately 3 years and that clock has started.
And then another question is about emergency use or compassionate use. And is there any plans to designate the drug for emergency use because most patients can't wait for the end of the study?
So that's a really important question, and it's something that's difficult to answer right now. And that's really because this is the first study. We've only assessed it in a few patients. So we don't yet know if the drug is safe or if it is effective, but it would be something that we would explore for patients should that be an appropriate avenue to take.
Question in the room? Are there plans to enroll children under 8 in the study?
Yes, so there are plans to enroll children under 8 in the study. So the way that the trial is designed is that we assess, particularly when we increase a dose level to a higher dose, we look to assess that in children or adults that are a little bit older. And then so the first few patients in a new cohort are always over 8 years old. And then once we've seen the safety profile in those patients, we then open it up to under 8s. But the study investigators at your site will be the ones who are up to date with where we are with enrolling patients and when under 8s can be included.
Is there a timeline or a year in which you anticipate the study would be completed?
So right now, based on our current projections of enrollment, we anticipate around 2025 is when we would get the first data for this study. We assess the safety as we go because that's really important for us to be able to, monitor that the patients are doing okay. But we won't have the complete data set probably until 2025.
Is there any chance that the method of administration of the drug would change?
So the method of administration won't change. And this is because antisense oligonucleotides have to cross what's called the blood-brain barrier. And your brain has this protective space that means that it's there so that it's not prone to any infection, because it's obviously a really important part of your body. So GFAP is produced in the brain, so we have to be able to get into the brain. And the only way to do that is to get into the CSF space. And that's why you have to do an intrathecal injection into the base of the spine. Now, there's a lot of work ongoing of how you can administer that in different ways. Right now we give it through an injection, but there's companies and other groups that are working to make that administration a bit more comfortable, but it would always have to be an intrathecal injection at this point in time.
Why are the initial patients being enrolled over 8? And when will the study begin enrolling patients under 8 or between the ages of 2 and 8?
So the reason that the study starts in patients that are 8 and older is when we go to regulators, like the FDA or, the European regulators, because this is the very first in-human study, so it's not being done in healthy volunteers, they want to make sure that they evaluate older age patients before bringing it into younger, particularly more vulnerable patients at younger age groups. And so 8 years old was just felt to be determined as the age in which the regulators were comfortable with us evaluating the drug in first. As I said, it all depends how quickly in a cohort we get those first patients in. We have a set number of patients that will be 8 or older, and then the rest of the cohort will open to under 2, but I can't give you a timeline of that right now because I just don't. We've only just begun enrollment and we're working through this in a safe and, methodical pattern. Yes. Mm-hmm. Yeah. So the first question was, how do we determine what the starting dose is? And then what did you say after that? Oh, yeah. And if there is potential for the doses to escalate. And then the second question was about if we know at this point in time what the shelf life is of the drug, i.e. how long can you keep it in the freezer for? So this is what's called a multiple ascending dose study. So we are looking at different dose levels. And the doses are determined based on the animal model data that we've seen and, what the knockdown looks like. So we start off usually at a lower dose that is still thought to be therapeutic. And then the next cohort receives a higher dose. And so we work through like that. And so we have strategies in place to increase the dose, but all along the way, we are looking at safety and making sure that at each dose level it's safe. And then second to that, we look to see, is it causing the reduction that we need? We don't have, when you run a clinical trial, you look at the stability of the drug. So we don't, and the shelf life of it. So we don't have that data now, but that work is always ongoing as we work our way through, because it takes quite a while to get that data, but it's something that we're working on. Any other questions?
Yeah, there are more questions in the chat. Katrina?
If a patient has a Baclofen pump, would the treatment be administered this way?
Is a Baclofen pump intrathecal? Amy, do you?
It is intrathecal, but I do not know we would not be able to administer drug that way because the Baclofen pump is going to be designed to release a certain amount of Baclofen into the space. And that we can't have that with this drug, right? We need the bolus, we need the drug to get in right away. So the Baclofen pump won't be able to be used as a conduit for it. There was also a sort of similar question about a shunt, talking about access.
Yeah.
So are there any patients receiving Spinraza, which is people might know is the drug for spinal muscular atrophy, who have a VP shunt? So obviously a different clinical program now. So are there any patients receiving Spinraza who have a VP shunt, or are there any thoughts regarding patients having a VP shunt and intrathecal administration of the medication, assuming Spinraza is similar to Ion 373?
Okay, so I don't know the answer to that off the top of my head, unfortunately. Particularly for this first study, the VP shunts are excluded. And I just don't have the answer to that question. I'm sorry.
Yeah, I mean, to talk a little bit about VP shunt. So a VP shunt has an opening from the spinal fluid into the abdominal cavity. So in theory, drug that is administered into that space would leak out. We don't know, or at least I don't know, Katrina, you might know, about the absorption and where the absorption would occur of the drug and whether the drug would leak out. it's a little too soon for us to answer those questions. Most children with spinal muscular atrophies don't have BP shunts because it's not as much of a brain disease as it is a disease that affects some of the big nerves and the neurons in the spinal cord and stuff. So it's a little bit of a different process. So there probably are not many patients that have both SMA and a VP shunt for other reasons, although I'm sure that could happen. There are a couple of additional questions in the chat about recruitment and processing and messaging. I don't know, Katrina, if you want me to read these directly or whether or not we want to address that a little bit. Is it easier if I read the questions? Katrina.
Okay. So I've been asked to explain the recruitment process into the trial. So the way that this works is that we have a number of sites identified globally. And this is because it gives us a good geographic distribution. When in a clinical trial, you know, we're looking to be as inclusive as possible. And so the investigators, as part of a clinical trial, you have inclusion and exclusion criteria. And so the investigators know the patients and their own patients the best. So really, they're the best people to make the decision of who would be a good candidate for a clinical trial. And so they take a number of different things into consideration, including, which obviously includes the inclusion and exclusion criteria. But it's based on investigators making the decision who is enrolled. Mm-hmm.
You said you make
Oh, this is a question about expanded access and compassionate use. So as I mentioned right now, we're in the early stages of the clinical trial. It is something that we would look to consider should the data emerge and be suggestive that compassionate use is a route to go down, but it's not something that is available right now. So the question is there data available from patients in cohort A? So we have a formal safety monitoring group. So they're responsible for monitoring the safety of the patients. And as I mentioned in my presentation, everybody that's involved in the study, including us at Ionis, are blinded to the data. The role of the data safety monitoring group is just to let us know that it's safe to proceed with the drug. So we haven't seen the data. We can't share that data because we're blinded, and it's really important for us to maintain the blind so that we're not biased in any way for what our drug is doing. But you can feel encouraged that at least the data safety monitoring group have seen the unblinded data, which they can't share with us, but they believe that the drug is safe and we can proceed with the trial.
The question was more about whether or not you could speak to whether anyone under 8 has been dosed.
Oh, yeah, there have been patients in cohort A that are under 8 years old that have been dosed. So remember, the trial right now goes down to 2-year-olds. So in cohort A, there were patients under 8 years old that were included.
So a couple more questions, and one of them involves some challenges we had this week about messaging to families that were coming from the sites versus coming from Ionis directly. Do you want to speak to that, Katrina, or do you want me to?
What was the question? Around sorry, you said messaging.
The question was that families had received communication that directed them to individual sites, such as CHOP, which was not consistent with information that I was providing about the trial. And so I guess wanted to just make sure that families understood there was a central way to get more information about the trial from Ionis, but that did not necessarily constitute a spot in the trial, even if you were directed to a specific site, that there seemed to be some a disconnect in terms of the messaging families were receiving, thinking that was a sign of hope that their child was going to be screened for the trial. And so I don't know if you want to speak to that, or if there's anything else that needs to be said about that website.
I can at least give our perspective. So Ionis is not involved in the recruitment process. It's not something that we're allowed to be involved in. So when sites are coming to us for information, we are there to give information about the clinical trial, you know, the design, point them in the direction of where centers are, where investigators are. We can't give any, we aren't allowed to recruit specifically into the trial. And then if patients are then being directed on to a center, it is up to the study center if they think, you know, they are still enrolling patients at their center. You know, if the trial participant is eligible for the study. So they're all factors that come into consideration. I don't know if you want to expand on that, Amy.
Yes, a little bit. I think that there are very few spots in Cohort B, just as this becomes more globally available. Yes, we aren't communicating directly with Ionis, but there is a central organizing committee that that all of us have to work through. So there is it's not Ionis and it's not the site. There's another a research organization that handles a lot of the scheduling and logistics for the trial. Yeah. Right. So yeah. I think there is a middleman between the sites and Ionis that does a lot of this. So I think there's some confusion in terms of when sites say, oh, we refer to Ionis or the sponsor, we're really talking more about that middle organization as we wait to get instruction from them. Whereas Ionis is saying it's up to the site. And so, you know, I think that's caused a little bit of confusion. So again, to an extent, the centers are operating independently and then working through this contracted research organization and awaiting further instruction in terms of next dosing and next spots from that organization so that they can organize the sites, and it's sort of a traffic control hub or something like that. So yeah, I don't know if that has caused some confusion as the sites are saying, you know, we're in limbo, or we're waiting for more instruction, and Ionis is saying it's up to the site. So just so everyone knows, that's sort of how things go.
Yeah, so we do, as Amy mentioned, we have a contract research organization, and they really assist us with the sites in terms of how to organize this clinical trial. And so they really help with, you know, looking at dates of when, you know, the whole teams are available at each site, making sure, just really assisting with the logistics. And so they help coordinate us all to try and make this as smooth as possible. But that is sometimes where there is maybe a little bit of disconnect in terms of communication, because sites are waiting on instruction from the contract research organization, and they just are really there to try and help us as much as they can.
So in terms of who is in charge of enrollment, and why are some sites more responsive maybe than others? I mean, I think some of the sites have had a little bit more experience having been part of Cohort A. So we sort of have a sense of how this is going. A lot more sites have been onboarded since then. Many of the sites are waiting to confirm with families, I think, until we are given further instruction from I'll just call them the middleman that helps with coordination. So I think a lot of the sites are waiting. But I think one of the take-home points that I've heard this week and that we're hearing today is that we need a little bit more responsiveness from some of the sites in terms of moving forward. So families have been reaching out, and there's a lot of uncertainty still. Katrina, do you want to take the question about countries where there is no site, such as South America? I mean, continents, obviously, but South America, Latin America, like there are obviously several places throughout Europe that don't have a site. Do you want to take that one?
Yeah, so it is always challenging when you design a clinical trial to figure out what countries you're going to include. And remembering again, this is a research study, it's an experiment. We're trying to limit the amount of risk to patients as much as possible. This is the first in human study. We don't know until that injection goes into a patient how it's going to respond in a human. So we want to minimize the risk as much as possible. We have tried our best as we can to get a good geographic distribution. So we have a number of centers in Europe. We have a number of sites in the US. You know, we have sites elsewhere in the world to try and be as fair as we can. I know it's immensely frustrating when there isn't a trial site open in a country that's near you, but really we've selected the countries and the centers because we think that we can get participants into this trial as quickly as we can and get to the answer that we're looking for, which is this drug safe and effective in as few people as possible, but also as quickly as possible. So should the drug work, we can then, you know, move on to the next step and rolling it out.
And just to add to that, you know, one of the things, you know, we're obviously testing the safety, but also the efficacy of this drug. And to determine the efficacy, we really need to know whether or not a patient can follow instructions. And so we really need them to be testing in their native language. It's very, even if someone speaks English, you know, traveling to another country where it's not necessarily their native language, you know, when we're asking them to follow commands or do certain things, you know, were they not able to do it because they didn't understand strength versus power, versus were they not able to do it because of the disease itself? So I don't know if that's fair to say, Katrina.
Yeah, that's absolutely fair to say. It's about minimizing the variability, and also, you know, a clinical trial is a huge burden. You know, we're asking people to come in and do a lot. And so I think, you know, traveling a long way is a huge ask for patients as well. And as Amy mentioned, you know, it would be impossible for us to have everything available in every language too. And so it's really trying to get to the answer as quickly as possible.
There was another question about, well, there's a question, do you have sites in Mexico? And the answer to that right now is no.
No, yeah.
If willing to travel wherever in the US, is there a chance to get the child in a site not close to us? Oh, okay. So some say still recruiting on clinicaltrials.gov.
Mm-hmm.
But one of the messages that I had given some patients was that we did not have openings for screening at CHOP.
Yeah.
That there were just a few patients identified, and we had already identified the patients that would be moving forward with screening at CHOP.
Yep. So clinicaltrials.gov reflects the study as a whole. So it's, you know, when it says recruiting, it means that all of the sites are actively open and recruiting. The sites have their own, you know, logistics that they have to operate around. They have a lot of patients that are coming to them, but there are only, you know, a certain number of spots available in this trial. And so I think your best course is if the investigator has told you that there aren't any spots available locally, then you should trust their guidance. You know, we're trying to work together to recruit to this trial as quickly and as effectively as we can. And I know that that means that some patients are going to be disappointed if they don't make it in.
Do you want to say anything about screen failures? I saw that somewhere in here. What happens if a site screens a candidate and the candidate fails the screening at that site for some reason?
So screen failures are a natural course of a clinical trial. You know, as I mentioned, we have inclusion and exclusion criteria. There are quite strict restrictions in place, and at a time, you know, even though something might pop up that just makes a patient ineligible and therefore they screen fail, it's up to a study center what their approach would be for screen fails. So it's based.
Yeah, I will tell you what my, you know, I think the only way to be fair about this at our own center if someone screens fails, but it does not delay the trial in any way,
Yeah.
then perhaps there would be a possibility to rescreen, but I cannot promise that I would hold up the trial for at least the 3 months that someone would have to wait. I don't think that's
Yeah.
fair to the community.
Yeah.
So if someone
exactly.
screens fails at my site, we would probably go to an alternate. So yeah, so I think, does the spot open back up at the site? I think that's, for my site, I don't think that it's fair to hold up. So I want people to understand up front that if they screen fail, there may not be another opportunity. There's some math here that I will share. How many patients are enrolled at this moment? Does every cohort have 42 divided by 3 or 14 spots?
That's not something that we can speak to.
Okay. I do want to revisit, while we're waiting to see if I caught all of the questions here. I do want to revisit the question about the drug being available before it is FDA, EMA, or regulatory approved. So going back through compassionate use or open access or the programs. So I've heard Ionis answer this question before. So Katrina, I've heard people say that as a company and organization, you do have information on your website about open access programs, but my understanding is that before something becomes available for such a pathway, you need to show that it works and is safe.
Yeah.
So
exactly.
just because a drug is in trial right now doesn't mean it's eligible for that pathway. So most of the time, and Katrina, like this is, I'm maybe a little out of my lane here, but most of the time my understanding is that if you have data from the trial but have not yet gone to the FDA, or you're waiting to go to the FDA, and things look promising and safe, that might be an opportunity. It's something that Ionis, I believe I've heard you say, is committed to that pathway if it's available, but it's too soon to know whether that would be.
So it's too soon to know right now for exactly the reasons that Amy highlighted. If the data suggests, things are moving in the right direction, we will explore the options of compassionate use or expanded access, but it is just something that we can't answer right now. We do not have enough data to be able to make that decision, but it is something that we're assessing as we go, and we're thinking about, but it's just not available right now.
So to follow up on that, the question is, so would that not happen until 2025? That I actually, not being from the pharmaceutical partner, I don't actually know when that could be available or an option.
We don't know either. It's all based on the data that comes in. It's difficult for us to be able to say right now.
But I think that's one of the reasons, for example, that I would not want to hold up the trial if somebody screen fails. I would not want to delay it another 3 months if there's other patients that could possibly move into that spot, so that we could keep things moving as quickly as we possibly can.
Yeah.
And you may not want to answer this one. How many spots are actually available for Cohort B?
I can't answer that question. I'm sorry. Do you want to do your session now? Are there any other burning questions or anything that's not been asked or some level covered?
For those who do not get into the trial, when can the community expect to have general access to the drug via a typical clinical setting?
I can't answer that question. I'm sorry. It's just something we don't know right now.
The only thing that I can say to that is CHOP is very committed and very forward thinking. If we are getting to a point where there is positive data and Ionis, not we, but Ionis is happy with those results and they're anticipating a positive review by the FDA, we would move heaven and earth to make this clinically available if given FDA approval, as quickly as we possibly can, working very closely with Ionis.
Yeah, our aim is to move this program forward as quickly as possible, but as safely as possible. I know that some of these questions might feel frustrating because we don't have the answer, but we are, as I mentioned, we are at the very beginning, and it's really tricky for us to be able to answer some of these questions, and that's just because we're just getting data right now and we're just at the start.
So while we're waiting for a few more, see if we've answered all the questions about the trial, because I'm not actually going to be speaking about the trial. I wanted to welcome Dr. Rusnikoff from Stanford. So you heard a lot of CHOP specific language today. I asked Dr. Rusnikoff as one of the site PIs if she also wanted to join us if there are some specific questions that she can answer. Unfortunately, some of the other PIs are in some of the other sessions at the ULF.
Yeah.
But I know, Mara, are you there, Dr. Rusnikoff?
Yeah, I'm here. Can you hear me okay?
Yeah, we can hear you.
Okay,
Hello.
perfect. Hi. Well, I just wanted to make a general comment, which is, I think a really tricky thing here is that even though Alexander disease is rare, there's a sizable number of children and adults with Alexander disease right now, and everybody's at a different stage of, relative urgency or change clinically. And we're so sensitive to that as providers, caring for your children and your families. And so something that's been probably the trickiest thing for all of us is that there are absolutely more perfect candidates for this trial than spots. The spots for each specific site are very limited, and there's a lot of kind of logistical kind of dancing around, timing and such that has to happen. And so, I think something that's been, in all honesty, very devastating for me and sad throughout this trial is that sometimes I think it feels like it's probably impossible to feel like you're getting as much transparency as you would want as families, because it's just so hard for us to know, or we're waiting for something. And so I hope that we can continue to answer your questions and get through this trial as soon as possible, because there's no question that there are going to be perfectly eligible families who won't make it into these first cohorts. And there's no question that that's so devastating and difficult. So I would just want to acknowledge that, because it's something we work really, really hard on behind the scenes.
Thank you so much for acknowledging that. Okay.
We do want to just remind everyone that, while we're very focused on the clinical trial, it is very important to me to echo what Dr. Rusnikoff just said, that first and foremost is taking care of the Alexander disease community. Yes, the trial is a wonderful opportunity. We're grateful to Ionis, and we're fortunate that we get a chance to bring this experimental therapy to our patients. But please understand that we are here for you regardless of whether or not you are in the trial or not. I don't know, Dr. Rusnikoff, if you want to answer that last question in the chat. I will address it for CHOP. And I will. What? Do you want to answer that, Katrina?
Oh, no. Just, we have the organizer here.
Oh,
Sorry, I rudely stepped in to grab the microphone. I just want to be cognizant of the time. Do you, so Amy, you're on. Do you want to kick off your section? Is this now a good time?
I think this is a good transition. I think I just want to address the last question in the chat.
Please.
So it's I think a little bit confusing between recruiting, screening, dosing, and all of these things that are part of this trial. In terms of recruitment, I will say CHOP is not actively looking for additional patients because we have so many patients that are eligible for the trial. In terms of working into this next phase of screening and dosing, that's the stage that we're at. We at CHOP have already identified for the few spots that we already have identified who we're moving forward with screening. And then unless there's a screen fail, which would open up another opportunity, I just wanted to share that transparency so that people are understanding where we are in the process.
And just to emphasize that is specific to CHOP and Amy's site.
Yeah, and I'll say for our site, we're a little bit more in flux at the moment because we haven't done our first screen for Cohort B at this time. And so while we're not necessarily, we've been contacted by enough families over the course of Cohort A to Cohort B that we're not currently, kind of actively looking for additional families. We have not yet notified with certainty who we will and will not screen for Cohort B.
Okay. Well, thank you all so much for all of your questions and allowing me the opportunity to speak to you. And thank you so much to Dr. Waldman and Dr. Ruznikov for also weighing in on this.
Thank you. Thanks for fielding all those questions. Wonderful. So Amy, I have you, or sorry, Dr. Walden, I have you on next. And so I'm going to transition. I just want to be cognizant of the time. Let's both stay connected so that we can get to our last speaker. Thank you.
And how much time do we have?
I believe we wrap up at 3:45 Central Time. And
Okay.
so is 20 minutes approximately okay for you?
Yeah, I actually did not plan a formal presentation. I actually just wanted to talk to the community, and I think this is a good time to transition. The reason I didn't prepare formal slides is because actually I was supposed to be on vacation. It's just a very busy time. But a couple of years ago when we started these gatherings, we started having yearly updates, even having one in person in Madison, Wisconsin several years ago. And we went to filling our Zooms, of over 100 people joining, to a call today where we have 39. So our numbers have dwindled quite a bit. So I wanted to spend some time trying to understand how we can help our families. I will be honest in saying that I feel that our community is more fractured. And I think part of that right now is obviously because of the pandemic, obviously because, I couldn't even be there in person today, and it's hard for families to travel, but we don't feel as tight as we once did. And I get a lot of families that feel that they're isolated or on an island, or feel that their child or their loved one with Alexander disease doesn't match what they're seeing online. So it doesn't match, maybe it's an adult or a teenager that's not a child, or maybe it's a child and not an adult. It's more the other way around. And so I don't feel that we're coming together as much as a community as we have in the past. And so I would love to have some open dialogue. It's a little bit weird because I'm staring at a podium and not necessarily staring at the people in the room. But maybe I can, at least on my computer, make this more gallery view, and really want to hear from you. What questions do you have? Do you want to have gatherings like this? Do you want to have webinars? Do you want to have things about symptoms? Do you want to hear about, research as that comes up? I will open the floor, I think, to questions and really want to understand what we need as a community and how we can more come together. I know some people feel very fortunate that they're part of the trial, and some people are very disappointed that they're not necessarily part of the trial, but wanted to try to understand how we can support each other.
I'll let you take the questions. It looks like you're looking at them right now.
Yeah.
Wonderful.
So it seems like one of the messages, I think, is that we shouldn't make all of our gatherings about the clinical trial. And I do understand that. I do agree with that. I think this particular meeting at ULF came right around the time of Cohort B launching and a lot of questions about Cohort B. So I think there was just some natural discussion about it and a lot of families having questions about it. But I do understand the point very well that we can't have all of our meetings be about the clinical trial. That's not fair to most of the Alexander disease community who will not have a chance to participate in the trial. Obviously, if there's an approved therapy, then that's a different story. Are people interested in formal presentations and webinars? Are people interested in more social gatherings by Zoom? Are people interested in being put in touch with more families that are like yours? So it seems like I'm getting some clinical updates and maybe some specific Alexander disease get-togethers. Are people Zoomed out? It's easier than traveling. And maybe some social hour chats. Do you want formal presentations? Do you want to just see the faces of other people who are experiencing what you are? So it seems like some question and answer sessions regarding symptoms. Our doctors here often can't answer my questions, and there's reliance on social media. So let me dive into that a little bit as we're going through some of these questions. So Dr. Resnikoff and I cannot give direct advice for an individual patient. And that's where it gets a little bit challenging. That being said, I do think that, having some general things, there was a question in the chat about whether Alexander disease itself causes pancytopenia. And what pancytopenia is low white count, low platelets, and low red blood cells. Alexander disease itself does not cause that, but drugs do. So a lot of the drugs that I use all the time in this disease can contribute to pancytopenia. So we can answer general questions. We can't necessarily say, you should have this done or you should have that done. We also have put together some information sessions for physicians. And I can sort of dig that up. That's through the Global Leukodystrophy Initiative. And it was something that we did at CHOP a few years back, where we spoke about Alexander disease. And so maybe we can provide some updates for physicians. We're also in the process of writing a number of papers that we hope would be helpful. And also making sure physicians know that they can contact us directly if they have questions. Informal meetings, I'm getting a lot of vibes, and we can also do both. We could do a couple of meetings where we have more symptom-based, or how you manage different symptoms of this disease, more information for families, but then also have some social hours or informal meetings where families, and maybe the physicians aren't even part of that, just for families to connect. Social media, I will just say a few things about social media. My recommendation is that people who are in the clinical trial do not post anything on social media. I don't think that's fair to all of the patients that wanted to be part of this trial and didn't get their chance. So I feel very strongly, and I also think that it threatens the integrity of the trial. In a different disease, not Alexander disease, I can identify by name, and I can identify their graphs and their trajectory of their treatment simply from social media. And so I think it's extraordinarily problematic in a placebo-controlled trial, right? So posting videos of your child walking or doing other things, or yourself, I think there's no place for social media in clinical trials, perhaps other than to announce that a clinical trial is up and running. But social media does allow us to connect, or Zoom or whatever does allow us to connect in these ways. So it seems like the general consensus is maybe some less structure, informal Q&As, round robin style, where everyone can talk about wins and losses in a support group type style. I think that sounds wonderful. So I'm gonna maybe lean a little bit on Elisa's corner and NDXD to maybe help us figure out how to organize those. And I think that would be a huge win for the community. And maybe they have a guest speaker like Dr. Messing, myself, Dr. Resnikoff, and maybe there's no guest speaker, and people figure out how they can connect without having to be formal in terms of the physicians.
Dr. Waldman, one thing that I've done with another group, and we can see if this sounds interesting to others too, is that we would have a panel of a couple of physicians and just really quickly talk about, like, let's say it's, treatment of neurological symptoms. You kind of talk about them a little bit, but then most of the session is for parents to ask questions, not patient specific, because of course we can't do that if we're not your child's doctor, but we can talk about more general things like, what do we tend to see, or, what more overarching questions that everybody can benefit from. And we did one with GI, one with neurology, one with genetics for another group, which I thought was kind of cool.
I will also take this opportunity. I'm not going to necessarily, well, you know what, let me share some slides real quick.
Dr. Walden?
Yeah.
This is Siobhan. If I can interject for a moment, I was curious, because I think it might be a nice transition, and perhaps that's what you're jumping into right now. But around managing clinical symptoms and creating a care team, are those two topics that you might be addressing in the next while you've got your time slot?
I wasn't going to present slides on those. Sorry for the phone ringing in the background. It was just important to me. Let me see if I don't know if I can share it. Is everyone seeing this?
We are able to see it.
I just wanted, I can speak to that a little bit. I really wanted people to understand, I'm just going to say it this way. There's a lot of charged political topics right now. It's very important to me that everyone knows that the NIH is funding work on Alexander disease, right? So I'm not going to touch the other political topics, but I just wanted just a reminder that the Global Leukodystrophy Initiative Clinical Trials Network is funded by the Rare Disease Clinical Research Network, which is an NIH initiative. And I just wanted families to know that, you know, there's a very broad mission statement of the goal of the GLIA network. But I just wanted to tell you a little bit about this. You know, so our GLIA program by the numbers, there's 8 clinical centers. Let me see if I can should close that. There's 8 clinical centers, 50 advocacy partners and growing, over 2,200 patients that have leukodystrophies. This is not related just to Alexander disease. And Alexander disease is one of the 30 disorders. And I just want everyone to know the participating sites in the US. So Stanford is one of our sites, Utah, Baylor, Children's CHO of Atlanta, Kennedy Krieger is shown here. Here's Children's National, MGH with Florian Eichler, and CHOP.
5 minutes. 5 minutes.
I just
For
wanted
those on the
everyone-
call, could you please mute your line? We're getting some background noise. Thank you, Dr. Walden. I'll let you go.
Oh, no worries, no worries. So we are working, and this is a research initiative where we're trying to look at outcome measures. So we're taking what we've modeled in Alexander disease and really extending it to all of these different sites. And so you don't necessarily have to travel from California to Philadelphia to be part of this initiative. So this is, we are continuing our study at CHOP. The studies are differing a little bit at some of these other sites, but they're still doing blood work and spinal fluid and other things that you've experienced when you've traveled to CHOP. So just something, you know, to share. I don't want to make this too much research oriented. Here are some of for our international patients on the call. There's around the world, there's some other external collaborators that may not be funded by the NIH, but are certainly our partners, everywhere from Canada through Australia, led by Dr. Fatimi, who we were just mentioning. Dr. Eichler on the left, who we were just mentioning, who I think just received an award, Dr. Fatimi at Kennedy Krieger and Dr. Vandiver at CHOP. And we are doing the biomarker project where we're looking specifically at novel biomarkers in Alexander disease. And this is looking at the measuring of GFAP in the spinal fluid and blood. So I just wanted everyone to know that our research has continued and that the NIH is really, your tax dollars are going to support research in Alexander disease, which I think is a lovely thing that's very important to me that everybody knows. Okay. So in terms of what we could talk about at a future meeting is thinking about symptom-based approaches to this disease. Some people don't think about certain symptoms being related to the brain, especially when it comes to the gut or the bladder or bowels. So those are things that are sort of hot topics that we can talk about. We can maybe have a physical therapist. I think that's a great idea that talks to different families about certain things. Again, we have to just be careful that it's not medical advice, that it's just general advice. And lots of ideas that we can go to. And then one of the things that I've talked to families about in the past is how to have a care team. You know, it's wonderful that you want to travel to Philadelphia or to Stanford to try to get to be seen by our teams, but you really do need to have a local care team, and we can provide some advice on how to do that. Okay.
Thank you. Are there any additional questions on the line that you see or in the chat? Do you have any questions? Okay. Just doing a room check here. I'm whispering, you know, through my mask, which I'm sure is not easy, but we don't have any.
Yes, and I agree. I think it's time to turn this over to Angel 8, and I actually do have to run, so I might have to leave just a few minutes early. I'm sorry that I'm going to miss Angel 8's presentation.
And something, just one moment, Dr.
Yeah. COVID vaccine? We do recommend the COVID vaccine for all ages with Alexander disease. You know,
Oh, I'll let you finish your thought. That wasn't the question, but I'll let you finish that sentence because I interrupted.
No worries, no worries. Yes, so we are recommending the COVID vaccine. COVID is just, even though it's migrated and Omicron and whatever the new variant, maybe that people aren't necessarily as sick, it still is the best way to protect yourself and to mitigate some of the symptoms and to lessen some of the symptoms. So we still strongly recommend the COVID vaccine. It does not affect your eligibility for the trial, but I will tell you that getting COVID during the trial for those patients that are participating or wish to participate is very, very difficult because we have to change the schedules. We have to cancel your appointment. You become off schedule. It is not a prerequisite for the trial, but it's something that we strongly recommend because we really don't want to be doing spinal taps without anesthesia, and anesthesia will cancel within 30 days at CHOP if you've had COVID. So it affects the schedule, it affects the integrity of the trial. So it's not a requirement, but it is recommended at our site. And yes, for children under 5, I do recommend the COVID vaccine.
We do too at Stanford.
Thank you for the clarification from both of you. And now here's the question.
Actually, this is Lee from NDXD, and it's not really a question other than I just want to offer us as the advocacy group, we would love to put together some of these Zoom meetings, some of these community, getting the community together around these topics, because I do think it's important. So please know I'm going to be reaching out to both of you and to others if other, a lot of the families that I'm talking to do have questions. And I think it's a great idea for us to get together over social media. So thank you for that.
Wonderful. That's part of this conference is bringing people together, making connections, utilizing people's expertise. Thank you for calling attention to that.
Thanks, everyone. Sending my best to all of you.
Thank you. Take care. And now I'd like to introduce someone that we have in our room here in just outside of Chicago. Here at the conference in person, we have Dr. Chelsea Haig. She's an artist, a researcher, and a coach, devoted to making space, witnessing, and telling the stories of families raising children today. She works at the Center of Educational Neuroscience, Visual Storytelling and Healing, and Parenting. Today, she will be our last presenter, so let's put our hands together as we transition. Thank you, everyone. We're just getting hooked up, so thanks for your patience. If you guys need to grab water, you have a few seconds, stretch a little. The perfect transition. And I didn't even read the whole thing. You've got quite a history here. That's okay. Where's Mills College?
Mills College is in Oakland.
Oakland, okay. I'm reading her bio.
Everyone, welcome.
My apologies. I'll let you take the mic and hook it up back to you.
I'll do that. Can you all hear me?
Okay.
Okay, I'm going to share my screen. Okay. Okay. Never mind. I'm not going to share my screen. There we go. Okay, so I am Chelsea. I'm so excited to be here with all of you today. And we're talking about some of the themes that were coming up as in this last conversation, right? So can I just pause for a second? I would love to be able to see folks' faces on my laptop since most people are here. Is there a way we can set it up so I can see everybody on my screen? Let's do that. I just want to be able to see you all. Oh, there's your beautiful faces. Much better. Great. Can you all see me too? Okay. So we're talking about some of these social components that came up. So caring for the caregivers, that's all of you. We're going to talk about the voice of the rare caregiver and of your community as we move through today. And I'm so excited to hear a little bit from each of you about what your experience has been like. So I'm Dr. Chelsea Haig. I work in visual facilitation and visual storytelling. And my colleague, Crystal, who's the founder of Angel Aid, is not here with us today. But she comes from a long line of work in the rare disease community, and we're just really thrilled and excited to be here with all of you today. So in terms of what we're doing, we're going to start off with a little bit of storytelling. I'm going to ask you all to participate in the conversation, share a little bit. I'm going to lead you through a couple of exercises. Then we'll talk about care in the community, the kinds of stressors that we typically see coming up for families like yours. And we'll finish off with what's going to be happening with our project that I will invite all of you into. So before I get, can you hear me if I put this down still? Are we all good? Before I get going, I want to say to those of you who are here that I know that you are all giving so much to the folks that you are taking care of. And you too are worthy of that amount of love, that amount of care, and that amount of support. And that is why we're here, right? We know that, yes, there is deep medical knowledge and concepts and work happening from all of your families, and there's a whole bunch of other stuff going on. And so really wanting to pause for a moment and acknowledge the journey that you've been on, acknowledge the space that you're in, and acknowledge all of those multiple things happening in your lives. So to that end, we're going to start off with a little exercise. So I want you to sit, if you're sitting or if you're standing, I want you to get somewhere where you're comfortable, right? Get both of your feet on the floor. Adjust yourself so that you're feeling good. And you close your eyes if you want. And I'm going to lead you through some exercises to calm our systems down, because one of the things that we know about this community and about medical trauma is that it activates our bodies so deeply that it's very hard to give and give and give and not also tend to ourselves. So close your eyes if you would. And we're going to start by taking both of your hands and reaching up behind your ears. And there's two little muscle groups up there. I want you to see if you can find them. And we're going to go down from behind your ears. The muscles go all the way to your shoulders. I want you to take 10 or 11 times to rub that, just stroking downwards, and play with it for yourself. You might be someone who likes really light touch. You might be somebody who likes firmer touch. What we're doing is activating parts of the brain to send you some calming vibes. So take a second and calm everything down. A second there. You can really hear me doing that when I have the mic here, huh? I'll move it. I won't, but you keep going. Okay. Let's take a deep breath together. I want you to take one hand and put it on your heart, and your other hand and put it on your belly. If it feels better, you can take one hand and put it on your forehead, and the other hand and put it on your heart. Whatever feels good and nurturing to you. And we're going to take 3 deep breaths here. Last one. I just want you to notice the feeling of the air around you, the sounds in the room around you, the feeling of your clothes on your body. I want you to notice how your body is feeling. So notice if you're carrying stress or tension in any of your muscles, shoulders, neck, hips, back. And see if you can identify the most relaxed muscle in your body and relax it just a little bit more. I want to move us now to an emotional check-in. So most of us carry in our bodies emotions, feelings that are either from the past or that are oriented to the future. I'm worried about X that happened last week. I don't know what to do about Y that happened last month. I'm thinking about getting my child into this clinical trial. I'm worried about something that's going to happen in the future. Just notice, with no judgment, what kinds of emotions or feelings are there. You may notice some that conflict with each other. You might not notice very many, and that is okay. Noticing what is there. And let's take one more breath together. Start to bring your awareness back into the room. Come on back onto the screen if you're on a screen. And I am curious for the folks on Zoom, and then I'll give the instruction to the in-people person here in just a minute, what that was like. And I want you to think about one feeling or one emotion that you identified as we went through that visualization. And in just a second, I want you to write it in the chat, but I don't want you to press send. So pull up your chat. Oops. Oh, how do I get the chat on here? And you can write it in. Where did the chat go? How strange. It's not, but I can just do that. Sure. And for those of you in the room, maybe we'll just go around the room. Oh, there's the chat. Great. We'll just go around the room and everybody can share just a single word. I'll start and then we'll go around this way. If you're in the Zoom, so all of you folks, you can go ahead and write your word in. And once we start sharing, you can press send and I will read them out. Okay. So I'll start and then we'll go around this way. You can participate too. Wow, you're so ready. Yeah, we're naming one emotion or feeling that we're having. Curious. You're on, my friend. Excited for those who didn't hear. Fulfilled. Forgiving. Calm. Oops, sorry. Centered. Calm. ¿Y tú, mi amor? Do you have one, sweetie? Good. Okay, and I would love to hear one word from those who are online with us. If you'd like to share, oh, my chat's back. If you'd like to share in the chat, you can either share directly to me or you can share with everyone. Calm. Okay, so that's open to you if you'd like to share as we move forward. And I will read them out. Acceptance, right? Yeah. Yeah, so lots of emotions and feelings come up, and I would imagine, aware that for many of you, they're multiple, right? There's more than just acceptance or curiosity or aware or calm. There are many. And at Angel Aid, that's what we focus on. We focus on working with you. So I loved to see in the chat earlier when Dr. Waldman was sharing about the kinds of social engagement and the kinds of support groups that you're hoping for. Because it really is through this kind of shared practice and being together that we can heal ourselves so that we can care for our loved ones. So Angel Aid is a global nonprofit network that provides folks connection and emotional support to rare families with somebody who's got a rare disease. initially founded for rare mothers. We do serve others now also who feel isolated and overwhelmed so they can experience compassion and relief. Our goal is to have 3.5 million rare families by 2026. That's all rare diseases. We don't differentiate, and we would love to see you in our community. We have quite a large network of folks coming together in a closed social network. So it's not, it's not a Facebook or an Instagram, but it is a private social network unique to rare families. So as we've done this work, what we've really seen when we talk to folks in a global survey that we did a couple of years ago, that when we talk to rare families, what comes up is that we need to see stories that are similar to mine. I need to know and to be able to feel that I'm not crazy, right? That folks need time and permission to do a little bit for myself. When you are consumed with caring for another, sometimes we need permission. We need someone to say, it's okay, you can care for yourself in connection, a listening ear without judgment. So at Angel Aid, we're not doing any fixing, right? We're an organization that witnesses and that will be with. So we do storytelling, self-care, and connection. And I want to share with you a little bit about the project that we have coming up that is specific to the Alexander community, specific to your group. But before we get there, I want to say a few words about self and community care and why it matters, why we are doing this work. So we know that there's a journey, right? There's a caregiver's map that cycles through hope and grief. And for those of you who are on, well, I think probably here in the room, it's a little bit small, but if you're on your own computer, you can probably read this. The cycle that we've identified most of our families go through, and I'll say this is not necessarily linear, right? Some families might cycle through again and again and again in a cyclical kind of way. So noticing changes, something's not right, looking for a diagnosis, seeking out support, adjusting to a diagnosis, learning about genetics, processing new information, dealing with overwhelm, dealing with fatigue, shifting responsibilities. So real strain around identity change and what does this mean? Learning about the disease, becoming an expert. Many of you on this Zoom were saying that you're the ones telling your doctors how to treat your family member. A need to learn nursing skills. Resistance sometimes to having to be the expert, to having to play that role as advocate or expert for your family member. Full-time care. So costs, loss of income, impact on relationship, impact on the family as a system as you navigate that. End of life, acknowledgement, preparation, bereavement, lots of denial and anger through that part of the process, and then survivorship, so real redefining of identity. And many families cycle through these, right? So though they're numbered, know that we expect that it's fluid. This is another way to look at it, right? So some people like to see it in this way. So it's again, noticing changes, adjusting to the diagnosis, a real increase in responsibilities, lots of caregiving, end of life, and survivorship. That psychosocial process is really where we focus at Angel Aid. So when we talk at Angel Aid about the kind of trauma that occurs as a family moves through a rare disease journey, we talk about CTSD, not PTSD. So PTSD is post-traumatic stress disorder, and CTSD is chronic, ongoing traumatic stress disorder, right? So it's an ongoing navigation of trauma that is present and that creates irrevocable changes in the lives of families. So the difference, and you can see here, right? It's like a stressful event is a one-time event and it comes down and there's some kind of baseline. Most of the rare families that we work with are caregiving, and traumatic stress is more of a moving target and it's always elevated. So there's shifts in the brain, right? Things happen in our brains and in our bodies when we're stressed like that in an ongoing way. So as we think about what we're doing and why we're here and the kinds of supports that the Alexander community needs, there for sure is a place for informational support, right? So that's many of the medical supports, education, tangible support, that's childcare or daily living tasks, folks coming into the home to provide physical supports. But there's also emotional supports and there's companionship supports. And that's really where we locate ourselves. So emotional support is about feeling seen, feeling validated, feeling understood. And companionship support is about engaging in activities with others, resulting in a sense of belonging. And let's be clear that both companionship support and emotional support move upwards, right? When you feel more emotionally supported, when you have a community of folks to relate to and to be seen with, you're much more able to do some of the caretake activities that are expected of folks. So what does Angel Aid do? And I saw in the comments lots of questions about support groups, about getting together, about social opportunities. And that's what we do at Angel Aid. So we offer ongoing support. We have the social network. We also have lots of different practitioners offering a multiplicity of kinds of psychosocial and emotional support on a weekly basis. So this is a quote from one of our rare moms. These mothers, rare mothers, sharing their stories, feelings, thoughts, concerns, fears, solutions, challenges, and hugs brings a degree of joy, solace, and peace to me that I have never felt before. This is a mom who had 3 children with a rare and fatal disease. I want to transition now to telling you a little bit about the project that we are going to embark upon, hopefully with all of you. And I'll read this bell hooks quote, "Rarely, if ever, are any of us healed in isolation. Healing is an act of communion." So when we think about that chronic traumatic stress journey and that elevation of stress hormones in the body and stressors on our relationships and on our families and on our children, what we're looking for at Angel Aid is a space to bring that down inside of community of folks that understand, right? That are part of your community. We are starting a project called Voice of the Rare Carrier. It's specific to the Alexander community. And this is the second time that we have had a community do this kind of project. So I'm an academic and an artist. And one of the things that I've seen with lots of different communities of folks, I've worked primarily with young adults who've had cancer, but also with parents with kids with special needs and a number of other kind of parent populations, is that when we can see our stories seen, visualized, and shared, it's really healing. And so I'm going to share a little bit about the last project we did, which was Voice of the Rare Carrier. And there's a lot of overlap here, right? So a lot of these folks were raising children that had rare diseases, not all of them, but they were all carriers of a rare disease. And we met 6 times. And each time we met, in our first meeting, we picked 5 topics to move through that were important to the community, that were most critically important to their experience of being rare carriers. And we'll do the same with your Alexander community. And then we're looking to storytell. So for 5 weeks, once a month, 5 times we come together for an hour, and it's an open space to share around a particular topic. And as we do that, I will draw it. Okay, so they're themes, right? You'll see some quotes in each of these. But they're themes that came out of this conversation. And this set of infographics that we create together as a community will be given to your physicians, your folks that work with you, companies looking to support you and looking to engage and work with you so that they have a better sense of who you are and how they can serve you. I am not a carrier of Alexander disease. And so as I draw these, I will bring them to you. And sometimes you probably will say to me, that wasn't it. And then we'll change it, right? The idea is for us to be in conversation about representation so that some of the stories and some of the challenges that are coming green. Can you all hear me again? Yeah, awesome. So we'll create 5 of these. This is the first one we created, and I'll share with you the second one. There's another one, right? And we're looking to share your stories, right? To hear what matters to you and to your families, and to share that with the folks that work with you in a way that makes sense, that is accessible, and that feels empowering to your families. So as we begin this work together, I'm so excited to invite you into this project. It will run in the fall, I think. We will certainly keep you aware. And I don't know, I'm not sure what's going to come of this. I certainly know that there will be some medical components, right? Some of this will be about your journey through the medical system, and some of it probably won't. And all of those things are okay. So as a facilitator, my role here is to make space for you to share your stories and to do some of the visual work to help them get represented in ways that feel good. So with that, I think I want to leave it there and open this up for questions, concerns, comments about what this will look like if you should choose to participate with us in the fall.